Hi all, welcome back to another day of Bio 148. We're going to be talking this week about genetics, and I think a lot of you all will have fun with this. A lot of people like this section, so I think you'll like it. The initial part of figuring out meiosis is kind of tricky, so we're going to do kind of a different type of bio connection than what we usually do, where it's a worksheet. Okay, so kind of like how the last one was a little different, this one is as well. Okay, so I will be starting to walk you through that as well, but for now, let's just get started with playing with meiosis and remembering things about homologous chromosomes. So let's get a new note started. Okay, back to my notes for the summer. All right, and I think today is day 16. So that's our new note. And day 16. Whoa, okay, my kids had this. There's a crayon, I didn't know that. Okay, cool. Let's go here. So day 16, and today we're going to be talking about homologous chromosomes. And meiosis. I decided I am going to do another video tomorrow talking about fertilization and mitosis. So just remember that is coming tomorrow, and I'll probably talk a little bit about gene expression again as well. Okay, so let's start by thinking about homologous chromosomes. So we've kind of talked about this before, but let's just do a reminder. So here I have my cell. Here in my cell, maybe I'm in G1. I've got the R gene, and I've got a capital version and a lowercase version. And then I also have a second set of chromosomes, and these ones have the B gene on them. Okay, so let's do a big B. Oops. Whoa. What did you do with my eraser outline? Okay. Okay, so let's do the B gene. And I like to put it in a different spot. Let's put it down here in the bottom. And the B gene. Okay, so let's remember some terminology here, okay? So here, these two things right here both contain the R gene. Okay, so we've got same gene in the same location. Can you go somewhere else with that, please? Okay, so same genes in the same locations on these two different chromosomes, okay? And when that happens, those are called homologous chromosomes. Okay, and here I have two different versions of the R gene, okay? So they can have different alleles. And when they do have different alleles, that's when we're going to call them heterozygous. Okay, so we can compare that, okay, to, actually I'm going to do green instead. These two chromosomes. So these chromosomes have different genes. And when they have different genes, they're non-homologous chromosomes. Okay, so remember that when we have the same genes and the same locations, those are going to be homologous chromosomes, right? When we have different genes on these different chromosomes, we're going to have non-homologous chromosomes. Okay, so this is how I can start my cell. And after this stage, so here I'm in like G1, okay, is also how the cell would look like in G0. And remember that these single lines that I'm drawing here, okay, are actually two DNA strands, right? So remember that I have two DNA strands per DNA molecule. And remember that these strands are going to separate during S phase, okay? So during, oh, I need some more. So during S phase, those strands are going to separate from each other. So here I have my R chromosome, my capital R, right? Here are my two different strands. I've got that R gene kind of being encoded, okay? That R allele being encoded on both of them. And so I'm going to have the new strands being made and my centromere, right? And so these two are going to be held together. So those original strands split apart, right? I'm going to use just like a light gray for that second strand, just so that you guys can see a little bit of a difference, okay? So we've got, oops. One old strand and one new strand per DNA molecule, right? So here, semi-conservative replication uses each strand of original molecule as template to make new strands, right? So don't forget that. And that's why we have this capital R being encoded on both sides, right? And the same thing is going to happen for my other homologous chromosome here. So those two strands are going to pull apart. I'm going to have those new strands being made, still my centromere. And in this case, I had, remember the little R, right? So I had that little R to start. Here now I've got, let's do it this way, actually. Oops. Just another way to show this, okay? Got that big B gene, right? The strands are going to pull apart from each other. Be used as a template to make the new ones. Oops. Okay, there we go. Okay, template to make the new ones. So I have that centromere, and so that B gene is encoded on both, right? Last one. Got the little b. Okay. Going to pull them apart, those two strands apart that are part of the same molecule at that point. Right, now we've pulled them apart to be two different molecules. We're going to use each one as a template. And so we have that little b gene on both, and we still have that centromere holding them together, okay? So remember how S phase goes, okay? So S phase, think synthesis, synthesize, it makes it, right? Think semi-conservative replication, right? So here I'm showing, where'd my pen go? Okay, here I'm showing, let's choose another color. A few additional things, right? So I still have these two, right, are still going to be homologous. Actually, let's change my color there. What did I do? Yellow, right? So this one and this one, these are still homologous chromosomes. The chromosomes are replicated now, okay, but they're still homologous to each other. And then I also have my non-homologous chromosomes. And I have some additional things now too. So here, two DNA molecules that are part of the same chromosome, okay, remember are called sister chromatids. And they are identical DNA molecules. that are part of one replicated chromosome, okay? So they're identical, they're going to have the same genes and the same alleles, okay? And that's a little bit different, right, than these two, right? So here I've got non-sister chromatids, and these are DNA molecules that are part of the two homologous chromosomes. Okay, so here we've got our homologous chromosomes, right, here and here, and here and here, right, you can see it. When we have a single DNA molecule from one chromosome and comparing it to another single DNA molecule from a different homologous chromosome, that's when I'm going to have non-sister chromatids, okay? So they're going to have the same genes, but could, right, have different alleles. So in this case, they do. Okay. All right, so we'll get back to what we call them when they have the same alleles in a couple of days. I just want you to think about them as having different ones for this exercise today. Okay, so now let's start going through the process of meiosis. So we did homologous chromosomes, another vocab, right? And now let's start playing with these. So I'm going to just take this cell to start, copy it, pull it down, okay? Let's clean this up because we don't need all the definition things right now. Okay, so here, right, this is in G1 phase. So in G1, right, I have 1, 2, 3, 4, okay, DNA molecules. And I also have 4 chromosomes, okay? There's 4 unreplicated. And then I want to ask myself the question of whether or not I can see homologous chromosomes. Okay, and so here when you see 2 versions of the same gene, okay, or 2, even if this was capital R, right? I've got the same gene, the R gene, on 2 different chromosomes, okay? That tells me that it's homologous. So yes, so anytime that I see homologous chromosomes, right, like these 2 homologous chromosomes, then that answer is going to be yes. And whenever that answer is yes, that's going to tell me that the cell is diploid, okay? So think diploid, like 2 of them, okay? So 2 chromosomes with the same gene, okay? Okay, so now let's go through S phase, right? So S phase is what we drew up here, right? I could draw it again here. So in S phase, right, I've got, after S phase, I should say, okay, I have my R, my little r, that's a little r, my big B, and my little b. Actually, let's go back. I don't really like how I did that. I'm going to draw them as 2 separate lines. I get a little bit lazy sometimes in not doing it, but I want you guys to practice this first. So, oops. Remember that big R is on both sister chromatids, right? And of this first chromosome, it's on both sister chromatids of the second chromosome here. So both of these are like chromosome 1 because they have the same genes, but we have 2 different copies of it. Then down here we have capital B's and we have the lowercase b's. Okay, so now this is like after S phase. So I've gone through S phase and maybe I'm in prophase 1. Okay, so note that I am calling it prophase 1 because we're going to have a prophase 1 and a prophase 2. Okay, so here I'm in prophase 1 and I'm going to end up asking the same questions. Now in prophase 1, you'll see that I didn't draw the 2 DNA molecules, 2 strands per DNA molecule in the same way because in prophase 1, it's condensed down, okay? So each one of these, okay, now represents 1 DNA molecule, or that's 2 strands, right, stuck together. So you can kind of compare it to this one up here where we drew both of those 2 strands together, right? And remember they're held together by all these hydrogen bonds, right? These guys aren't splitting apart very easily, but they can, right? There's a whole bunch of hydrogen bonds holding them together, okay? Now, additional things happen, like histones will help out with this and other proteins to condense them down. So here I'm just showing it as a single line is 1 DNA molecule, but remember that it is still 2 strands, okay? So we're going to ask these same questions, okay? So I'm going to ask about the DNA molecule number. I'm going to ask about the number of chromosomes, and I'm going to ask about homologous, whether or not they're present, and then that will help me determine if it is something that we call haploid or diploid, okay? Okay, so here, when I'm looking at this, my molecule count now is 1, 2, 3, 4, 5, 6, 7, 8, right? 8 DNA molecules, but I've got 1, 2, 3, 4 replicated chromosomes, and I still have homologous chromosomes. I'm going to say yes to that. Oops. Oh, it wants to do math for me. Okay, I don't know how to turn that feature off. I'll have to try later. And then it's diploid, right? Because I answered yes, so it's diploid. Okay, so now the next thing that we need to think about is what happens next. And this goes back to that PMAT, right? So here I did prophase. So the next thing, remember, is going to be metaphase, okay? So next I'm going to go into metaphase 1. And in metaphase 1, remember, in metaphase, things align. In metaphase of mitosis, the sister chromatids aligned on either side of the metaphase plate. But here in meiosis, we're going to have homologous chromosomes aligning on either side of the metaphase plate. So I'm actually going to copy all of this to play with. And I'm going to move it around so that you guys can kind of see it. Okay, let's get rid of all these little dots and things. We're going to recount here in a minute anyway. Okay, so when I've got this going on, I want to add a little metaphase plate down the middle. Okay, and then I'm going to move these around so that they are on either side, homologous chromosomes. Whoa, that didn't do what I wanted. Okay. Let's not take that one. Okay. So here I've got homologous chromosomes on either side of the metaphase plate. Okay. So that needs to be a little R. It needs to be red so we can track it a little bit easier. I don't know why I changed it. Okay. So here we go. Let's move that guy down just a little bit. So what you'll see here is that I have a homologous chromosome, right? Homologous chromosomes on either side of the metaphase plate. So there's homologous chromosomes and here's homologous chromosomes. Also note, okay, what color was that I did? Non-homologous. Also note that there are both non-homologous chromosomes are found on the left side of the metaphase plate and on the right side of the metaphase plate, okay? Okay, so now I'm going to ask these same questions that I asked earlier. Copy those, bring them down. Okay, so let's get rid of all that. Okay, so my number of DNA molecules hasn't changed. I'm still at 8 DNA molecules, okay? I'm still at 4 chromosomes. I still see homologous chromosomes, so I'm still diploid, okay? Then in anaphase, telophase and cytokinesis, they're going to just separate further, okay? So let's just draw out anaphase quick, okay? So an anaphase, kind of going to be pulled by their belly buttons. Okay, so I've got that R. Oops, I forgot that I was still doing this. Headers, I guess. Okay. Okay, so here they're being pulled away in anaphase, but nothing else has changed, right? So here they're just being pulled away from each other, but all of this is still there, okay? So if I'm in anaphase 1, I've still got all of these things, okay? In anaphase 2, or sorry, in anaphase 2, some different things are going to be pulling apart, okay? But for anaphase 1, the thing that is being pulled apart, right, is, I understand, Evelyn, is the homologous chromosomes, okay? So now we're in telophase. We're going to change the shape of our cell a little bit is all we're going to do. So in telophase 1, a lot of that stays the same, but they're starting to be really far away, so I can even pull these apart even further and further and further. Right, they're being pulled apart. Okay. All right, so then after that, let's close up that cell. Okay. So then after that, we have cytokinesis, and they split into 2 separate cells. So after meiosis 1, okay, you would also think of this as prophase 2, okay, I'm going to have 2 separate cells. I'm actually going to get rid of that line up there because at telophase I haven't split yet. Okay, so after cytokinesis, I'm going to have those non-homologous chromosomes, 1 non-homologous chromosome in each cell. Or sorry, I don't know what I just said. We've got 2 chromosomes, 1 of each non-homologous chromosome in each cell. Okay, so over here on the left, I've got the capital R. On the right, I've got the little r. On the left, I've got the big B. On the right, I've got the little b. Okay? Okay, so now when I'm going through here, after meiosis 1 and prophase 2, right, when I'm at this stage, I can ask the same set of questions. Let's get rid of that. Okay, so now if I'm looking at each cell, okay, so this information is for each cell. Okay, so here I have 1, 2, 3, 4 DNA molecules, but I have 1, 2 replicated chromosomes. But here, note that even though I have sister chromatids, I don't have homologous chromosomes, okay? So now I have no homologous chromosomes, so I'm at a haploid state, okay? So at the beginning of prophase 2, I'm at a haploid state. Now from here on out, we're just going to go through the same thing that we would expect for mitosis, okay? So I'm going to copy all of that. Get rid of all this. Okay, so now I'm in metaphase 2. And the only thing that really changes here is going to be that I have this like metaphase plate starting to form. And it's going to be dividing the sister chromatids. Okay. So the metaphase plate there is going to be dividing those sister chromatids. So here, if I'm looking for my different numbers of DNA molecules, it's still the same. The metaphase plate is there, but there's no other change. Okay. Okay. So then I start pulling away anaphase, telophase, cytokinesis. Okay. So I'm not going to do all of the different, these different questions for actually those intermediate stages because I don't want you to worry about them, okay? So here I've got things pulling away, right? So here's kind of what anaphase could look like. Okay, so this could be like anaphase 2. If I'm in telophase 2, I'm going to be even further apart, right? And have the cell starting to kind of really separate. Okay. Okay, so I'm going to have the cell separating a lot more because I'm in telophase. Right, these are going to be going even more to the edges, pulled apart. Okay, so this would be at like telophase 2. Okay, and then after that I'll have cytokinesis, okay? So in cytokinesis, I'm going to end up with 4 total cells here. Oh, that one's too long. Okay, each one will have a long chromosome that has the R gene on it. Sometimes it's a capital R allele, sometimes it's a lowercase allele. The short chromosome is going to have the B gene, sometimes a capital B, sometimes a lowercase b, okay? And now we can ask these same questions again, okay? So after meiosis 2, we have what we call gametes. And remember that these are sperm or egg, okay? So these are going to be your sperm or your egg, and we can ask these same questions that we did before. So we can ask the number of DNA molecules, we can ask the number of chromosomes, we can ask whether or not there's homologous chromosomes, and we can ask if they're haploid or diploid. Okay, so here, number of chromosomes, 1, 2. Number of chromosomes, 1, 2, but they're not replicated. 2 unreplicated, okay? Homologous is no, and so they must be in their haploid state. Note, though, that we still have, what color did I do those? Green, right? Let's go back up. Yeah, I still have these non-homologous chromosomes, okay? So I have both non-homologous chromosomes. In these cells, okay? So the next thing that I want you guys to do is try to apply this. I'm going to be giving you an example of Sophia's gametes. We're going to be playing with 3 genes on 3 chromosomes, and I want to see how you guys can play with that and make it into your own to make it so that you can really solidify this idea and understand how things could line up. One thing that I do want to note is that up here where we had these guys, oh, not there, lined up this way in metaphase 1, we didn't have to line them up this way, okay? So we could have lined them up. I'm just going to copy it and pull it down. Okay, actually, no, I'm just going to show you quick and then I'll switch it back, okay? So I didn't have to line it up so that the capitals were on the same side. It could have been instead that I had the capital R's on the left and the lowercase b's, okay? And then the capitals over here. So I could have lined it up that way instead. I'm going to switch it back because I don't want you guys to get confused if you go back and look at these notes. But just note that I could line it up either of those ways on my in my cell, okay? And that's actually called something, okay? It is called the law of independent assortment, dependent assortment, and we'll talk more about that later when you get to genetics, and it's going to make it so that you can line these up in different ways, and it creates a lot of variation being possible in your population. Okay. All right, so that is it for now, and good luck with this. Have fun playing with Sophia's gametes, where it just kind of takes this to the next step, and let me know if you guys have questions. We'll come back and talk about fertilization tomorrow, and I'll do the learning objectives at that time. Okay, just focus on really understanding this, especially this haploid diploid thing really throws people off sometimes. So keep in mind that in these cells that are either egg or sperm, you're going to have both non-homologous chromosomes but no homologous chromosomes, right? Okay, so just kind of keep that in mind, what you have and what you don't have at this point. Okay, I will see you all again tomorrow. Good luck, have fun, and let me know if you have any questions that pop up as you go.